In this video, we're going to talk about the structure of proteins. And what basically is a protein? A protein is a polymer consistent of many amino acids. So each amino acid represented by the circle is a monomer that forms the protein. Whenever you have many amino acids, it's also called a polypeptide. And the bond that connects each individual amino acid residue is a peptide bond. So let's talk about the individual structure of an amino acid. So an amino acid has a chiral carbon with a hydrogen attached to it, and it has an amine group attached to it as well. And then it has an R group and a carboxyl group. Now I'm going to draw another amino acid right next to it. The left side, where the nitrogen is located, is the N-terminal of the amino acid. The right side, that has the carboxyl group, is the C-terminal of the amino acid. Now these two amino acids can react with each other in a condensation reaction where we're going to lose water, and we're going to connect the two amino acids into one molecule. So once water is removed, we could form a peptide bond. This is also called a dehydration reaction because you're losing water. And so this is going to be the new product of the reaction. So let's see if I can fit it in here. So this oxygen is gone. So now we have a nitrogen. Let me take out H2O. We have one hydrogen left over. We lost two once we lost water. So this is a dipeptide because we have two amino acids combined. And so this amide bond that we see here, that is the peptide bond. We formed a bond between the carbon and the nitrogen atom. So whenever you combine two amino acids to form a peptide bond, you're forming a covalent bond, which is hard to break. Now let's talk about the different levels of protein structure. There's 4 levels that you need to know: the primary structure, the secondary structure, the tertiary structure, and finally the quaternary structure. Now the primary structure is simply based on the sequence of the amino acids found in the protein. The sequence determines the shape of the protein. If you replace just one amino acid with another, it will completely change the shape of the protein. So the shape and its function is primarily determined by the sequence of amino acids. The secondary structure describes the localized shape of a protein. And there's 2 of them that you need to be familiar with: the alpha helix and the beta pleated sheet. In the alpha helix, you could see that it's stabilized by hydrogen bonds. The NH group of one amino acid interacts with the carbonyl group of another amino acid. And each turn contains about 3.6 amino acid residues. Now here is a visual illustration of the beta pleated sheet. Just like the alpha helix, it too is stabilized by hydrogen bonds between the carbonyl group of one amino acid and the NH group of another amino acid. Next up, we have the tertiary structure, which represents the 3-dimensional complete folding pattern of the protein. And so here's a visual illustration of it. So we could see some areas is just a straight chain. Here we have an alpha helix, and here we have a beta pleated sheet. Now, the tertiary structure is one individual subunit. When you combine multiple subunits, you create a quaternary structure. So hemoglobin is an example of that. It has 4 individual subunits, 2 alpha subunits and 2 beta subunits. So hemoglobin is a protein with a quaternary structure. And so that's basically it for this video. Hopefully it gave you a basic understanding of proteins and the 4 levels of structure that they have. Thanks for watching.